Hello students, today we will learn about virus resistant plants. How transgenic plants are used as virus resistant plants. Okay, so we will start now. Virus resistance. Virus infections of crops may result in retarded cell division, that is hypoplasia, excessive cell division, that is hyperplasia, and cell death, that is necrosis. The overall effects of virus infections are growth retardation, lowered product yield, and sometimes complete crop failure. The chemical methods used to control various plant pathogens will be ineffective with respect to plant viruses since the viruses are intracellular obligate parasites. There are, however, certain safe agricultural practices to control or reduce viral infections to plants. Use of seeds that are virus free. Control insects that spread plant viruses. Control weeds that serve as alternate host for viruses. Use cultivars that possess virus resistance. It is possible to minimize plants against viral damages by expressing viral proteins in the plant cells. With the advances made in genetic engineering, it has become a reality to develop transgenic plants with virus resistance. This is mostly done by employing virus encoded genes, virus code proteins, movement proteins, transmission proteins, satellite RNA, antisense RNAs, and ribozymes. In recent years, some attempts are also made to provide virus resistance to plants by using animal genes. Now, we will discuss some developments. And first development is virus code proteins. The virus code protein mediated approach is the most one successful one to provide virus resistance to plants. It was in 1986 transgenic tobacco plants expressing tobacco mosaic virus code protein gene were first developed. These plants exhibit high levels of resistance to tobacco mosaic virus TMV, that is in short TMV, full tobacco mosaic virus. Excited by this remarkable success, scientists have worked with many more viruses, around 30, and developed crops with virus code protein mediated protection. The transgenic plant providing code protein mediated resistance to virus are rice, potato, wheat, tobacco, peanuts, sugar beet, alfalfa, etc. The viruses that have been include alfalfa mosaic virus, in short ALMV, cucumber mosaic virus, that is CMV, potato virus X, that is PVX, and potato virus Y, PVY, citrus trichosia virus, that is CTV, and R rice strip virus, RSV. Now let's know about the advantages of virus code proteins. The code protein genes from one virus sometimes provides resistance to some other viruses which may be unrelated. Example, TMB of tobacco plant provides resistance to potato virus X, alfalfa mosaic virus, and cucumber mosaic virus. Now let's know about the limitation of virus code protein. The virus code protein mediated protection is successful for viruses with single stranded RNA genomes. However, this approach is of not much use of viruses with genomes containing double stranded RNA and single stranded DNA. Now let's know about the mechanism of action of virus code protein. As a transgenic plant expresses the gene for code protein of a given virus, the ability of the same virus to infect the plants again is drastically reduced. Despite a remarkable success in the virus code protein mediated protection, the molecular mechanism of the protection is not clearly known. Now let's know about the movement proteins. As the virus infects, the plant cells, its rapid spread through intracellular junctions, that is plasmodesmata of vascular tissues, occurs through the participation of movement proteins produced by the viruses. Good examples of movement proteins are 30 kilodalton protein of tobacco mosaic virus and 32 kilodalton protein of Brome mosaic virus (BMV). Transgenic tobacco plants that express a mutated 30 kilodalton movement protein have been developed. The TMV infection to these plants is much less. It is believed that the mutated movement protein competes with the wild type TMV coded protein, thereby reducing the spread of the virus. In recent years, a recombination movement protein have the components of golden mosaic virus and African cassava mosaic virus have been developed. This protein effectively interferes with the spread of both viruses. The advantages with movement protein strategy is that it is applicable to single standard DNA viruses or Gemini viruses. Also, now let's know about transmission proteins. There is a good coordination and interaction between plant viruses and insect viruses for the spread of viruses from one plant to another. Certain viral coded and transmission proteins do this job effectively. It is possible to produce mutated transmission proteins and block the spread of viruses. Thus, the spread of insect transmitted viruses can be prevented by engineering crops to express a defective virus transmission proteins. Now let's know about the satellite RNAs. Plant viral satellite RNAs are small RNA molecules that multiply in the host cells with the help of specific helper viruses. These satellite RNAs are encapsulated together with the respective helper viruses. In general, the presence of satellite RNAs reduces the severity of the viral disease and the symptom and thus reduces the effective of viruses. Transgenic plants containing satellite sequences have been developed to provide resistance to plant diseases. One example is now we'll study. When cucumber mosaic virus CMV infects pepper plants, severe symptoms appear. These symptoms can be minimized with higher plant yield when CMV is co-inoculated with a satellite RNA. Satellite RNA approach is not widely used due to several limitations. Some of the satellite RNA may increase the severity of disease symptoms in some plants. Satellite RNAs mutate very rapidly, which may sometimes result in a highly virulent agent. Recombinations between satellite RNAs have been detected. This may lead to serious consequences. Now let's know about antisense RNAs. The antisense RNA approach is designed to specifically interfere with virus applications. By use of genetic engineering, a complementary DNA strand of a gene DNA sequence can be inserted in reverse orientation, and this is referred to as antisense gene. The mRNA produced by antisense gene is complementary to the mRNA synthesized by normal gene. As a result, both these mRNAs hybridize, and thus the normal transition of mRNA is blocked. The net effect of employing an antisense gene into a cell is that it blocks a specific gene expression. It is possible to introduce viral antisense genes into plants and produce mRNAs complementary to viral sequences involved in virus application. The antisense mRNAs can block the replication of viruses. Initially, antisense RNA approach was carried out in single-stranded RNA viruses. The success of this approach, however, was limited, probably due to some reasons. Let's know about the reasons. High concentration of antisense mRNAs may be required. Protein association with mRNA interferes with hybridization between sense mRNA and antisense mRNA. Antisense RNA approach may be more useful for DNA viruses. In fact, tomato golden mosaic virus insert TGMV replicase coding sequence was cloned in antisense orientation and introduced into tobacco plants. The transgenic tobacco plants expressed antisense RNA of TGMB replicase. These plants were resistant to TGMB infection. Now let's know about ribozyme. Ribozymes are small RNA molecules which promote the catalytic cleavage of RNA for providing virus resistance ribozymes in the form of antisense RNAs capable of cleaving the target viral RNAs have been developed. The ribosome binds to a small sequence of viral RNA and splices. In this way, it is possible to block the replication of viral RNA. However, ribozyme's approach has not been very successful in plants. Now, let's know about fungal and bacterial diseases. The plants do possess general defense systems against invading pathogens. This is, however, not truly comparable with the immune system of animals. Whenever there is a cellular damage caused by pathogens, that may be fungi or bacteria and plant waste, the general defense system of plants get geared up to provide some amount of protection to the plant. This natural disease resistance of plants is inadequate. However, knowledge on the natural systems of plant resistance is useful for the biotechnological approaches to develop disease resistance. Now let's know about pathogenesis related proteins. To defend themselves against the invading pathogen, plants accumulate low molecular weight proteins which are collectively regarded as pathogenesis related proteins. The different types of PR proteins and their properties now we'll discuss. First is chitinase. Chitin is a constituent of fungal cell walls which can be hydrolyzed by the enzyme chitinase. Certain chitinase genes from plants have been isolated and characterized. A bacterial chitinase gene obtained from a soil bacterium that is Serratia morsensis was introduced and expressed in tobacco leaves. Some other workers isolated a chitinase gene from bean and developed transgenic plants of tobacco and Brassica napus with this gene. The transformed tobacco plants were found to be resistant to infection of the pathogen Rhizoctonia solani. In case of beta napus, the protection, however, was comparatively less. Now let's know about glucanase. Glucanase is another enzyme that degrades the cell wall of many fungi. The most widely used glucanase is beta 1,4 glucanase. The gene encoding for beta 1,4 glucanase has been isolated from barley, introduced and expressed in transgenic tobacco plants. This gene provided good protection against soil-borne fungal pathogen Rhizoctonia solani. The resistance to fungal pathogens is much higher if both chitinase and glucanase producing genes are present in transgenic plants. By this approach, fungal resistant tobacco tomato, and carrot have been developed. Now let's know about the ribosome inactivating proteins. In short, RIPS. Ribosome inactivating proteins offer protection against fungal infections. They act on the large RNA of eukaryote and prokaryote ribosomes and thus inhibit protein biosynthesis. Certain RIPs that do not inhibit plant ribosomes were identified and the corresponding genes have been used to develop transgenic plants. Example, type 1 barley RIP is used to provide resistance to fungal infections. Some authors use the term antimicrobial proteins to RIPs. The other examples of antimicrobial proteins are lectins, deficiency, lysozyme, thionines, etc. Now let's know about lysozyme. Lysozyme degrades chitin and peptidoglycan of cell wall, and in this way fungal infection can be reduced. Transgenic potato plants with lysozyme gene providing resistance to Escherichia carotivora have been developed. Now let's know about defensins. Defensins are antimicrobial peptides found in all the plant cells. They attack the microbial plasma membrane. However, this is not adequate to provide resistance to pathogens. In recent years, an artificial defensin gene has been developed and introduced into potatoes. These potatoes developed resistance to the bacterium Escherichia carotivora. Now let's know about thionines. Thionine proteins also offer protection against bacteria. Thionine coding genes have been introduced into tobacco and transgenic plants so developed sold resistant to Pseudomonas syringae. Now let's know about phytoalexins. What do you mean by phytoalexins? Phytoalexins are secondary metabolites produced in plants in response to infection. They are low molecular weight and antimicrobial in nature. The phytoalexins, usually present in specialized cells or organelles, are mobilized when infection occurs. Further, during infection, there occurs induction of genes for increased production of phytoalexins. Stilbene synthase is a key enzyme for the synthesis of a common phytoalexin. The gene coding stilbene synthase has been isolated from peanut and introduced into tobacco, rice, and brassica nappas. The transgenic plants carrying stilbene synthase gene were resistant against some fungi. Now let's know about nematode resistance. Nematodes are simple worms found in the soil. They possess a simple digestive tract. The annual crop loss of the world due to nematode, that is roundworm infestation, is very high. Some workers have identified and cloned a nematode resistant gene from wild beet plants. It is proposed that this gene encodes a protein that detects the pest nematode and triggers a defensive reactions in the plant. It is believed that some chemical compounds that destroy the gut of the nematode are produced. Attempts were also made to transfer the nematode resistant gene to sugar beet. Not much success was reported, the major limitation being the difficulty in cultivating the gene altered cells of sugar beet. Now let's know about the resistance to abiotic stresses. Plants are constantly being subjected to environmental stresses that may result in deterioration of crop plants and a very low or even no yield. Plants are dependent on the subtle internal mechanisms for tolerance of various stresses. The in situ tolerance of crop plants, whenever present, is inadequate and therefore cannot give protection against the stresses. A wide range of strategies are required to engineer plants against a particular type of stress tolerance. Some of the abiotic stresses and recombinant strategy developed to overcome them we will now discuss. Now first we will discuss about herbicide resistant. Weeds, wild herbs. Weeds means wild herbs are unwanted and useless plants that grow along with the crop plants. Weeds compete with crops for light and nutrients besides harboring various pathogens. It is estimated that the world's crop yield is reduced by 10 to 15% due to the presence of weeds. To tackle the problem of weeds, modern agriculture has developed a wide range of weed killers, which are collectively referred to as herbicides. In general, majority of the herbicides are broad spectrum as they can kill a wide range of weeds. A good or an ideal herbicide is expected to possess some characteristics. Let's know about the characteristics. Capable of killing weeds without affecting crop plants, not toxic to animals and microorganisms, rapidly translocated within the target plant, rapidly degraded in the soil. None of the commercially available herbicides fulfills all these criteria. The major limitation of the herbicides is that they cannot discriminate weeds from crop plants. For this reason, the crops are also affected by herbicides, hence the need to develop herbicide resistant plants. Thus, these plants provide an opportunity to effectively kill the weeds by herbicides without damaging the crop plants. Now, let's know about the strategies for engineering herbicide resistance. A number of biological manipulations, particularly involving genetic engineering, are in use to develop herbicide resistant plants. First point we will discuss. First point is overexpression of the target proteins. The target protein being acted by the herbicide can be produced in large quantities so that the effect of the herbicide becomes insignificant. Overexpression can be achieved by integrating multiple copies of the genes and by using a strong promoter. Second point is improved plant detoxification. The plants do possess natural defense systems against toxic compounds, that is herbicides. Detoxification involves the conversion of toxic herbicide to non-toxic or less toxic compound. By enhancing the plant detoxification system, the impact of the herbicide can be reduced. Now, let's know about detoxification of herbicide by using a foreign gene. By introducing a foreign gene into the crop plant, the herbicide can be effectively detoxified. Mutation. Let's know about mutation of target protein. The target protein which is being affected by the herbicide can be suitably modified. The changed protein should be capable of discharging the functions of the native protein but is resistant to inhibition by the herbicide. Once the resistant target protein gene identified, it can be introduced into the plant genome and thus herbicide resistant plants can be developed. For success in the development of herbicide resistant plants, good knowledge of the target protein and the action of herbicides is required. Now let's know about glyphosate resistant. Glyphosate is a glycine derivative. It acts as a broad spectrum herbicide and is effective against 76 of the world's worst 78 weeds. Glyphosate is less toxic to animals and is rapidly degraded by microorganisms. In addition, it has a short half-life. The American chemical company Monsanto markets glyphosate as Roundup. Let's know about the mechanism of action of glyphosate. Glyphosate is rapidly transported to growing points of plants. It is capable of killing the plants even at a low concentration. Glyphosate acts as a competitive inhibitor of the enzyme 5-enol pyruvyl cyclimate 3-phosphate synthetase, in short EPSPS. Again telling 5-enol pyruvyl cyclimate 3-phosphate synthetase. This is a key enzyme in cyclimic acid pathway that results in the formation of aromatic amino acids, that is tryptophan, phenylalanine and tyrosine, phenols and certain secondary metabolites. The enzyme EPSPS catalyzes the synthesis of 5-enol pyruvyl cyclimate 3-phosphate from cyclimate 3-phosphate and phosphoenol pyruvate. Glyphosate has some structural similarity with the substrate phosphoenol pyruvate. Consequently, glyphosate binds more tightly with EPSPS and blocks the normal cyclimic acid pathway. Thus, the herbicide glyphosate inhibits the biosynthesis of aromatic amino acids and other important products. This results in inhibition of protein biosynthesis. As a consequence, cell division and plant growth are blocked. Further, the plant growth regulator indole acetic acid, it is an auxin, is also it's an auxin, that means like a hormone. Okay, so auxin is also produced from tryptophan. The net result of glyphosate is the death of the plants. Glyphosate is toxic to microorganisms as they also possess cyclimate pathway. Glyphosate is non-toxic to animals, that is including humans, since they do not possess cyclimate pathway. Of the 3 aromatic amino acids synthesized in this pathway, tryptophan and phenylalanine are essential and they have to be supplied in the diet, while tyrosine can be formed from phenylalanine. Now let's know about the strategies for glyphosate resistance. There are 3 distinct strategies to provide for glyphosate resistance to plants. First point is overexpression of crop plants EPSPS gene. An overexpressing gene of EPSPS was detected in petunia. This expression was found to be due to gene amplification rather than an increased expression of the gene. EPSPS gene from petunia was isolated and introduced into other plants. The increased synthesis of EPSPS by about 40-fold in transgenic plants provides resistance to glyphosate. These plants can tolerate glyphosate at a dose of 2-4 times higher than that required to kill wild-type plants. Now let's know about the use of mutant EPSPS genes. An EPSPS mutant gene that conferred resistance to glyphosate was first detected in the bacterium Salmonella typhimurium. It was found that a single base substitution C to T resulted in the change of an amino acid from proline to serine in EPSPS. This modified enzyme cannot bind to glyphosate and thus provides resistance. The mutant EPSPS gene was introduced into tobacco plants using Agrobacterium ti plasmid vectors. The transgene produced high quantities of the enzyme EPSPS. However, the transformed tobacco plants provided only marginal resistance to glyphosate. The reason for this was not immediately identified. It was later known that the cyclimate pathway occurs in the chloroplast while the glyphosate-resistant EPSPS was produced only in the cytoplasm. This enzyme was transported to the chloroplast, hence the problem to provide resistance. This episode made scientists to realize the importance of chloroplast in genetic engineering. In later years, the mutant EPSPS gene was tagged with a chloroplast-specific transgenic peptide sequence. By this approach, the glyphosate-resistant EPSPS enzyme was detected to freely enter chloroplast and confer resistance against the herbicide. Detoxification of glyphosate. Let's know about detoxification of glyphosate. The soil microorganisms possess the enzyme glyphosate oxidase that converts glyphosate to glyoxylate and amino methyl phosphonic acid. The gene encoding glyphosate oxidase has been isolated from a soil organism, Ochrobacterium anthropi. With suitable modifications, this gene was introduced into crop plants, example, oilseed rape. The transgenic plants were found to exhibit very good glyphosate resistance in the field. Now let's know about use of a combined strategy. More efficient resistance of plants against glyphosate can be provided by employing a combined strategy. Thus, resistant, that is mutant, EPSPS gene in combination with glyphosate oxidase gene are used. By this approach, there occurs glyphosate resistance as well as its detoxification due to glyphosate oxidase gene. Now let's know about phosphinothrene resistant. Phosphinothrene is also a broad spectrum herbicide like glyphosate. Phosphinothrene is more effective against broad-leaved weeds but least effective against perennials. Now let's know about the phosphinothrene, a natural herbicide. Phosphinothrene is an unusual herbicide being a derivative of a natural product, namely bialaphos. Certain species of Streptomyces produce bialaphos, which is a combination of phosphinothrene bound to two alanine residues, forming a tripeptide. By the action of peptidase, bialaphos is converted to active phosphinothrene. Now let's know about the mechanism of action of phosphinothrene. Phosphinothrene acts as a competitive inhibitor of the enzyme glutamate synthase. This is possible since phosphinothrene has some structural similarity with the substrate glutamate. As a consequence of the inhibition of glutamine synthase, ammonia accumulates and kills the plant cells. Further disturbances in glutamine synthesis also inhibits photosynthesis. Thus, the herbicidal activity of phosphinothrene is due to the combined effects of amino toxicity and inhibition of photosynthesis. Now, let's know about the strategy for phosphinothrene resistance. The natural detoxification mechanisms of phosphinothrene observed in Streptomyces species has prompted scientists to develop resistant plants against this herbicide. The enzyme phosphinothrene acetyl transferase of Streptomyces species acetylates phosphinothrene and thus inactivates the herbicide. The gene responsible for coding phosphinothrene acetyl transferase, that is BAR gene, has been identified in Streptomyces hygroscopicus. Some success has been reported in developing transgenic maize and oilseed rape by introducing BAR gene. These plants were found to provide resistance to phosphinothrene. Now let's know about sulfonyl ureas and imidazolin linones resistance. The herbicides namely sulfonyl ureas and imidazolinones, inhibit the enzyme acetolactate synthase, that is in short ALS, a key enzyme in the synthesis of branched chain amino acids, namely isoleucine, leucine and valine. Mutant forms of this enzyme and the corresponding genes have been isolated, identified and characterized. Transgenic plants with the mutant genes of ALS were found to be resistant to sulfonyl ureas and imidazolinones, example maize, tomato, sugar beet. Now let's know about resistance to other herbicides. Besides these, some other herbicide resistant plants have also been developed, that is bromoxynil, atrazine, phenocarboxylic acids, cyanamide. It may, however, be noted that some of the herbicide-resistant transgenic plants are at field trial stage due to environmental concern. In few of these plants withdraw, example atrazine-resistant crops. Now let's know about the environmental impact of herbicide-resistant crops. The development of genetically modified herbicide resistant crops has undoubtedly contributed to increase in the yield of crops. For this reason, farmers, particularly in the developed countries like USA, have started using these genetically modified crops. Thus, the proportion of herbicide-resistant soybean plants grown in USA increased from 17% in 1997 to 68% in 2001. The farmer is immensely benefited as there is a reduction in the cost of herbicide usage. It is believed that the impact of herbicide-resistant plants on the environment is much lower than the direct use of herbicides in huge quantities. There are, however, other environmental concerns. First point is disturbances in biodiversity due to elimination of weeds. Second point, rapid development of herbicide-resistant weeds that may finally lead to the production of super weeds. Tolerance of water deficit stresses. The environmental conditions such as temperature, that is heat, freezing, chilling, water availability, that may be shortage due to drought, and salinity influence the plant growth, development and yield. The abiotic stresses due to temperature, drought and salinity are collectively regarded as water deficit stresses. Now let's know about the causes of water deficit. Water deficit may occur due to some reasons. First is reduced soil water potential, increased water evaporation in dry, hot and windy conditions, high salt concentration in the soil, that may be decreased soil water potential, low temperature resulting in the formation of ice crystals. Now let's know about the effects of water deficit. Results in osmotic stresses, inhibits photosynthesis, increases the concentration of toxic ions, reactive oxygen species within the cells, loss of water from cell causing plasmolysis and finally cell death. Tolerance to osmotic stresses. The plant cells are subjected to severe osmotic stress due to water deficit. They, however, produce certain compounds collectively referred to as osmoprotectants or osmolytes to overcome the osmotic stress. Osmoprotectants are non-toxic compatible solutes and are divided into two groups. First is sugar and sugar alcohols, example mannitol, sorbitol, pinetol, ononitol, trehalose, fructans. Now second is zwitter ionic compounds. These osmoprotectants carry positive and negative charges, example proline and glycine butane. The production of a given osmoprotectant is species dependent. The formation of mannitol, proline and glycine butane are more closely linked to osmotic tolerance. Now let's know about the strategies to develop water deficit tolerance plants. As explained above, osmoprotectants offer good protection to plants against osmotic stresses and therefore water deficit. It is therefore logical to think of genetic engineering strategy for the increased production of osmoprotectants. Some progress has been made in this direction. The biosynthetic pathways for the production of many osmoprotectants have been established and genes encoding key enzymes isolated. In fact, some progress has been made in the development of transgenic plants with high production of osmoprotectants. Transgenic plants with glycine butane production. Glycine butane is a quaternary ammonium compound and is electrically natural. Besides functioning as a cellular osmolyte, glycine butane stabilizes proteins and membrane structures. Some of the key enzymes for the production of glycine butane have been identified: choline monooxynase, choline dehydrogenase, butane aldehyde dehydrogenase. The genes coding these enzymes were transferred to develop transgenic plants by using choline oxidase gene from Arthrobacter species. Transgenic rice that produces higher glycine butane, which offers tolerance against water deficit stress, has been developed. Now let's know about the resistance against ice nucleating bacteria. Formation of ice on the plant cells is a complex chemical process. The importance of ice nucleating bacteria is recognized in recent years. The occurrence of these bacteria has been reported in most of the plants, cereals, fruits and vegetable crops. The ice nucleating bacteria synthesize proteins which coalesce with water molecules to form ice crystals at temperature around 32 degrees Fahrenheit. As the ice crystals grow, they can pierce the plant cells and severely damage the plants. Now let's know about the chemical treatment of plants to protect the ice formation. Plants can be treated with copper containing compounds to kill the bacteria. Another approach is to use urea solution so that the ice formation is minimized. Let's know about ice minus bacteria to resist plants from cold temperature. Bacterium Pseudomonas cereus is one of the high prevalent ice forming organisms in nature. With genetic manipulation, gene that directs the synthesis of ice related bacterial proteins in P. syringae was removed. These newly developed bacteria are referred to as ice minus bacteria. The researchers proposed to spray the transgenic ice minus bacteria onto young plants. The intent was that these bacteria would give frost tolerance to the plants and thus increase the crop yield. The opponents of DNA technology were against this approach, the main fear being that bacterial mutants may create some health complication in humans. The researchers argued and justified that no new genetic information is introduced into P. syringae and it is closely related to all aspects to the parent one which is already in the environment. After prolonged court proceedings in USA, clearance was given for spraying the ice minus bacteria in the field. It was in 1987 ice minus bacteria were sprayed onto the field of potato plants and strawberry plants. Another strain of P. syringae commercially labeled as frostbian was later developed and used in crop fields. It may be noted that ice minus bacteria of P. syringae were the first transgenic bacteria that were used outside the laboratory. Fortunately, the experiments yielded encouraging results since crop damage due to frost formation was found to be reduced. Arabidosis. Now let's know about Arabidosis with cold tolerance genes. Scientists were successfully in developing cold tolerant genes around 20 in Arabidosis when this plant was gradually exposed to slowly declining temperatures. They also identified a coordinating gene that encodes a protein which acts as a transcription factor for regulating the expression of cold tolerant genes. By introducing the coordinating gene, expression of cold tolerant gene was triggered and this protected the plants against cold temperature. More work is in progress in this direction. Now let's know about the improvement of crop yield and quality. With the advances made in plant genetic engineering, improvement in crop yield and quality have become a reality. The crop yield is primarily dependent on the photosynthetic efficiency and the harvest index. The quality of the crop is dependent on a wide range of desirable characters, nutritional composition of edible parts, flavor, processing quality, and shelf life. Now let's know about green revolution. Green revolution led by Borlaug Swaminathan, whose enabled the world's food supply to be tripled during the last 3 decades of 20th century. This was made possible by adopting genetically improved varieties of crops coupled with advances in crop management. The development of high-yielding varieties of wheat and rice has enabled several developing countries to move from a position of food scarcity to become net exporters of these cereals. The Green Revolution became a reality as the farmers adopted a new cereal seeds. Besides employing high input method of agriculture, use of nitrogen fertilizers, herbicides, pesticides, modern equipment of agriculture, etc. Now let's know about the selected examples of crops for quality and yield. There are a wide range of crops that have been manipulated by scientists for improved yield and quality. Now let's know about genetic engineering for extended shelf life of fruits. Let's know about genetic engineering for extended shelf life of fruits. The genetic manipulation of fruit ripening has become an important commercial aspect in plant genetic engineering. Delay in fruit ripening has many advantages. It extends the shelf life, keeping the quality of the fruit intact. Long distance transport becomes easy without damage to fruit. Slow ripening improves the flavor. Genetic engineering work has been extensively carried out in tomatoes and some of the development we will discuss. Biochemical changes during tomato ripening. Fruit ripening is an active process. It is characterized by increased respiration accompanied by a rapid increase in ethylene synthesis. As the chlorophyll gets degraded, the green color of the fruit disappears and a red pigment lycopene is synthesized. The fruit gets subtained as a result of the activity of cell wall degrading enzymes, namely polygalactoronase, and that is in short PG, and pectin methyl esterase. The phytohormone ethylene production is intimately linked to fruit ripening as it triggers the ripening process of fruit. Addition of exogenous ethylene promotes fruit ripening while inhibition of ethylene biosynthesis drastically reduces ripening. The breakdown of starch to sugars and the accumulation of a large number of secondary products improves the flavor, taste and smell of the fruit. 3 distinct genes involved in tomato ripening have been isolated and cloned. The enzymes encoded by these genes and their respective roles for ripening will now discuss. Let's know about the genetic manipulation of fruit ripening. Scientists have been trying to genetically manipulate and delay the fruit ripening process. Almost all the attempts involve antisense RNA approach. Now let's know about the manipulation of the enzyme polygalactoneuronase. Development of flavor sour tomato. As already we discussed, softening of the fruits is largely due to degradation of cell wall, that is pectin, by the enzyme polygalacturonase PG. The gene responsible for PG, the rotting enzyme, has been cloned. PTOM6. The genetic manipulation of polygalactoneuronase by antisense RNA approach by the development of flavor sour tomato. Now we will discuss. It is by Calgene company in USA. First point we will discuss. First point is isolation of DNA from tomato plant that encodes the enzyme polygalactoneuronase. Transfer of PG. PG means galacturonase. Transfer of PG gene to a vector bacteria and production of complementary DNA molecules. The introduction of complementary DNA into a fresh tomato plant to produce a transgenic plant. Now let's know about the mechanism of PG antisense RNA approach. In the normal tomato plant, PG gene encodes a normal mRNA that produces the enzyme polygalactoneuronase that is actively involved in fruit ripening. Complementary DNA of PG encodes for antisense mRNA which is complementary to normal mRNA. The hybridization between the sense and antisense mRNAs renders the sense mRNA ineffective. Consequently, no polygalactoneuronase is produced, hence fruit ripening is delayed. Now let's know about the rise and fall of flavor sour tomato. The genetically engineered tomato known as flavor sour by employing PG antisense RNA was approved by US Food and Drug Administration on dated 18th May 1994. The FDA rules that flavored sour tomatoes are as safe as tomatoes that are bred by conventional means and therefore no special labeling is required. The new tomato could be shipped without refrigeration to far off places as it was capable of resisting drought for more than 3 weeks. Double the time of conventional tomato. Although flavor sour was launched with a great fanfare in 1995, it did not fulfill the expectation for some reasons. Transgenic tomatoes could not be grown properly in different parts of USA. The yield of tomatoes was low, the cost of flavor sour was high. It is argued that the company that developed flavor sour in its overt enthusiasm to become the first biotech company to market a bioengineered food had not taken adequate care in developing the transgenic plant, and unfortunately within a year after its entry flavor sour was withdrawn and it is now almost forgotten. Now let's know about the manipulation of ethylene biosynthesis. It has been clearly established that ethylene plays a key role in the ripening of fruits. The biosynthetic pathway of ethylene is now we will discuss. Ethylene is synthesized from S-adenosyl methionine via formation of an intermediate, namely 1-amino cyclopene 1-carboxyl acid, catalyzed by the enzyme ACC synthase. The next step is the conversion of ACC to ethylene by ACC oxidase. Three different strategies have been developed to block ethylene biosynthesis and thereby reduce fruit ripening. We'll discuss more next in we'll discuss more in next video. Any questions regarding this, you can ask me in the comment box. Thank you. Please subscribe my channel to know more updates about biotech topics. And thank you.